genetics. You see, when a mommy and daddy love each other.
Whoa!
Morning students of biology, Miss Cytosine here. Today I will be talking about interphase, mitosis, meiosis, and cancer. So to begin, does anyone know the correct order of the cell cycle?
Is it G1, G2 synthesis and mitosis?
Actually, the correct order is G1, synthesis, G2, and M.
Correct. Caitlin, the first stage is G1, in which the cell is growing. This is followed by the S phase, or synthesis, where the DNA of the cell replicates. And then in G2, the cell continues to grow until finally it replicates in the meiotic phase. After interphase is done, cytokinesis, which is right there, is the division of the cytoplasm and allows cell division to be completed. In animal cells, the cleavage furrow forms, and in plant cells, a cell plate is formed. The cell cycle is regulated by various checkpoints that tell the cell to arrest or to proceed the cycle. The G1, for example, the G1 checkpoint tells the cell to proceed with the S phase, or to enter the G0 phase where the cell does not divide. Protein kinase is also important in regulating the cell cycle. Can anyone tell me a name of a protein kinase that aids the cell cycle?
Cyclin-dependent kinase?
Awesome. And Caitlin, can you tell me how it helps the cell cycle?
It becomes active when attached to cyclin and in sufficient concentrations. Allows cells to move past the checkpoint.
Great job, guys. Moving on to cancer, there are two different types of tumors. A benign tumor is a mass of abnormal cells that doesn't affect the functioning of neighbor cells. A malignant tumor is also a mass of abnormal cells, but it divides uncontrollably and invades the functioning of neighboring tissues and organs. Also, a growth factor is a protein released by a cell that signals other cells to divide, and it contributes to the density inhibition of cell division. Does anyone know what the spreading of cancerous cells from the origin of the site to other tissues and organs is called? Okay, well, it's called metastasis. Remember that. Going on to mitosis, the whole point of it is that the cell divides and ensure that each new cell has the exact same genetic information. An easy way to remember all the phases is IPMAT, which is interphase, prophase, metaphase, anaphase, and telophase. Does anyone know what happens in prophase?
Chromosomes become visible and the mitotic spindle forms and the nuclear envelope disappears.
And are the duplicated chromosomes chromatids or chromatins?
Chromatids.
Excellent. Moving on, the next phase is metaphase. This one is simple. The microtubules of the spindle attach and the chromosomes are lined up on the metaphase plate.
Is the next phase telophase?
Nurses seriously needs to get her stuff together, okay? Everyone knows that after metaphase is anaphase. Doesn't she know it, Matt? Miss C, the next phase is anaphase, right?
Yes, exactly. In this phase, the replicated chromosomes are split in the centromere and move farther away, all the way to the opposite poles of the spindle. The last phase of mitosis is telophase, where the small nuclear vesicles of the cell begin to form once again around the group of chromosomes at each pole. Here is a diagram that summarizes all of mitosis. All right, now before we get all up in meiosis, let's talk general sexual reproduction. Narissa, can you tell me how sexual reproduction allows for the introduction of genetic variation?
I don't know why Miss Cytosine thinks I had the answer. Like, did I have my hand up? Well, through crossing over an independent assortment.
Yes, perfect. Now, differences between a haploid cell and a diploid cell. Caitlin?
A haploid cell is created through the process of meiosis, only has one set of genetic information, meanwhile a diploid cell has two sets and undergo mitosis to produce more diploid cells.
What types of cells are diploids and which are haploid?
Somatic cells are diploid and gametes are haploid.
Amazing, guys. Now, the difference between meiosis 1 and meiosis 2 is that meiosis 1 is the first division that occurs in which a diploid germ cell splits to create two haploid cells. Meanwhile, meiosis II is the second set of divisions that occur in which two haploid cells are created that were created in meiosis I undergo meiotic division to create four haploid cells. In prophase I, DNA coils into chromosomes, the nucleolus and the nuclear envelope disappear, and the meiotic spindle forms, and crossing over occurs. What is the meiotic spindle?
It's the region of a chromosome where the microtubules of the spindle attach to.
Actually, it's what equally divides the chromosomes during both types of division.
Correct, Narissa. We now have recombinant chromosomes because of crossing over, so the chromosomes have genetic information from two sources. Before I go on, something I forgot to mention, a zygote is the result of fertilization, and sex chromosomes are the pair of chromosomes that determine the biological sex, male or female.
And there are 22 autosome pairs and one sex chromosome pair.
Yeah, Caitlin, but next time you wish to speak, raise your hand. Okay, going on to metaphase I, the tetrads randomly line up along the midline and the spindle fibers attach to each centromere of the homologous chromosome. Oh, by the way, what's a tetrad?
It's the homologs that form during the crossover in prophase I.
Perfect, Caitlin. Now in anaphase I, homologous chromosomes move to the opposite poles of the cell, and in telophase I, the chromosomes reach the opposite end of the cell, and cytokinesis begins. Any questions? Moving on to meiosis II, in prophase II, the spindle fibers form and begin to move the chromosomes toward the midline of the dividing cell.
What are spindle fibers again?
Spindle fibers are the same thing as the meiotic spindle.
Oh, gotcha.
The same thing happens for the rest of meiosis II that happened in meiosis I, but it results in 4 new daughter cells.
Man, that was long.
I know, right?
The process of mitosis is happening in our body every second that passes, but meiosis is different in both genders. Females are born with their eggs, but hormones produced in puberty allow the process to finish. Meanwhile, males, the gametes are produced once puberty has started and never stops.
I guess you could say the testes never get tired.
Man, that was a good one, Marissa. Oh, something I forgot to mention is the RAS gene and P53. RAS is involved in the signals passed between cells that control the amount of growth that is allowed at any time. Cancer-causing mutation of RAS genes creates a form of the protein that is always on. P53 is a gene that codes for a protein that regulates the cell cycle and therefore functions as a tumor suppressor. Alright guys, it has been a pleasure teaching you guys today, and have fun with Miss Adenine tomorrow.
Thank you.
Hey, Miss Adenine here. Today we're learning about DNA replication. Let's start off with what makes up the DNA.
A DNA molecule is shaped like a double helix and is made up of a 5-carbon sugar molecule, a phosphate group, and a nitrogenous base.
Yeah, typically deoxyribose and 4 nitrogenous bases, named adenine, thymine, cytosine, and guanine.
Aren't those your teachers' names?
Don't call out.
Where did that come from?
If you look at each of the sugar phosphate backbones, you see that the 5' carbon has a phosphate group attached to it, and the 3' carbon has a hydroxyl group. This asymmetry gives a DNA strand a direction. Okay, so can anyone tell me the base pairs for a DNA molecule?
The hydrogen bonding between guanine and cytosine, and the hydrogen bonding between thymine and adenine.
Okay, anyways, these base pairs can create base sequences. Okay, these base sequences can make up a DNA molecule. So, let's move on and talk about what makes up RNA.
RNA also has a sugar phosphate backbone, but instead has ribose as its sugar, and instead has thymine as one of the nitrogenous bases, while uracil is a base that binds with adenine.
Also, RNA is single-stranded, unlike the double-stranded DNA.
Nerissa is such a great student. It's always great to have an enthusiastic student. Okay, let's finally begin talking about replication. DNA replication is a process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. We begin the process of DNA replication with the enzyme helicase. Can anyone tell me what helicase does?
Helicase unwinds
The double helix splicing the hydrogen bonds between the base pairs.
Great job. The point where the splitting starts is called the replication fork. Okay, the unwounding sections can be used to create two complementary DNA strands. The lagging strand forms in the opposite direction of the replication fork. Unlike the leading strand, which goes in the direction of the replication fork. Like Nerissa correctly stated.
Ay, como no puedo creer que Narisa está actuando como preferida del profesor. Como like,
After the leading and lagging strands are formed by the enzyme DNA polymerase III, a RNA primer called RNA primase helps begin the new DNA chain. The RNA primase adds new nucleotides to the new chain continuously from the 5' to 3', adding to the 3' ends. This is why the RNA primase is only used for the leading strand and not for the 3' to 5' lagging strand. Tell me what the lagging strands are made out of that causes this 3' to 5' direction.
Okazaki fragments.
Correct, Okazaki fragments, which are made from small bits of RNA primer that gives the dipole 1 a starting point on the strands. Dipole 1 then gives the small RNA primers off the lagging strand. The enzyme DNA ligase then helps bring the Okazaki fragments together.
isn't there another DNA polymerase that works during DNA replication?
Wow, yes, there is, and it's called DNA polymerase 4. Dipole 4 proofreads to make sure there's no mismatched bases at the end of a strand. Okay. Now let's look at the centrifuge diagram. Two scientists concluded that DNA replication follows a semi-conservative model by comparing their result to the results predicted by each of the three models shown here. The first replication of the N14 medium produced a band of hybrid, which included N15 and N14 DNA. This result eliminated the conservative model. A second replication produced both light and hybrid DNA. As a result, that eliminated the dispersive model and supported the semi-conservative model. Okay, well, that's basically it for DNA replication. Any questions?
How are you such a great teacher?
I just am. So.
Hi, my name is Miss Kalani. Let's start with a little genetics joke. What did Gregor Mendel do when he founded genetics? Whoopee!
What is she talking about?
Who's Mendel?
Really? So, anyways, the man to remember, Mendel. He was an Austrian botanist who discovered the basic principles of heredity by crossing-breeding pea plants. By crossing pea plants, he made two laws. The law of independent assortment, which states that alleles of two or more different genes get sorted into gametes independently of one of another.
And the law of segregation states that paired alleles segregate independently, right?
That's right, Angie.
Pea plants? Why pea plants?
Good question, Charlize. He used pea plants because they can make many offspring, and they have distinct phenotypic characteristics, which will help when he crossbreeds them.
Oh, I get it. Thank you.
She doesn't get it. She would just let Miss G continue so she doesn't get any more confused. I know Shirley's.
I actually didn't, but I just wanted to let her continue.
So what Mendel did first was that he crossed purple flowers with white flowers. We call this the pea generation.
How did he cross them?
He used a paintbrush to transfer the pollen from one plant to another ring.
That's right, Angie.
She's such a know-it-all. Like, she thinks she knows everything. Besides, no one needed to know that anyways. Wait, is that gonna be on the test?
So when he did this, all the offspring, or F1 generation, as we call it, was purple. What type of cross do you think this is?
Dihybrid?
No, but good guess. It's a monohybrid because one gene is being observed.
Obviously.
Later on in the lesson, we will talk about dihybrid crosses in which two genes are being observed. Now, in a monohybrid cross, one gene is being observed, so each homozygous parent in the P generation produces only one kind of gamete. So when, for example in this, when a yellow and a green is crossed with each other, it makes a heterozygote, which is in the F1 offspring, which produces two kinds of gamete, hence lowercase y and uppercase y. And then in F2, when they're breeded together, another heterozygote, it creates a 3-to-1 phenotypic ratio in which 2 will be heterozygote and 1 will be homozygous recessive. And that will be in the F2 population. So now we're going to talk about the Hardy-Weinberg equilibrium, which detects the frequency of a genotype in a population. Now, a way to calculate the frequencies of the alleles in a population is by using the Hardy-Weinberg principle. Now, P squared would be dominant, Q would represent homozygous recessive alleles, and 2PQ would be heterozygous, and that should all equal 1 if you add them together. Now, when you do a Punnett square will help you figure out the frequencies and the probabilities of getting each genotype. Now, in this one here, if you cross a homozygous recessive with a homozygous dominant, you get all heterozygotes. Hence, all of them have the same genotype, which is uppercase R, lowercase r. Now, an example in a problem, say, let's say a dominant allele D confers normal length fingers, and recessive allele lowercase d confers a short middle finger. If 9 out of 100 individuals in a population have short fingers, hence lowercase d, 2 of them, the frequency is 9 out of 100, or 0.09. Since the double lowercase d equals q squared, then q just equals 0.3. And since p plus q equals 1, and knowing that q is 0.3, you can calculate and figure out that p is 0.7. So, Charlize, what you were talking about before with the dihybrid cross, here is a dihybrid cross in which two genes are being observed. So in P generation, in this example, this one is not only yellow, but it's also round. As opposed to this one, it is going to be wrinkled and green. So, the genotype for something that is round, it would be an uppercase R and another uppercase R. As opposed to yellow, it would be uppercase Y and uppercase Y. Now for green, that would be lowercase y and lowercase y, because that is dominant recessive, and this was dominant, this is homozygous dominant. And well, this one, and then for wrinkles, it would be homozygous recessive. Now, when they mix these together, there are more, as you can see, there are more different types you can get. So the phenotype distribution ratio would be 9 to 3 to 3 to 1. Because as you can see here, that since there are two genes that are being observed, there's more genetic variation. Another thing that you will be talking about tomorrow was thymine. So this is an example of the dihybrid cross. Okay, we're good. So, any questions?
Like every time. What do you think of it?
Good. Complete dominance is when the dominant allele completely matches the recessive allele. As a result, the plant would show the complete phenotype of the dominant allele.
So if a heterozygous plant has a dominant allele for red and a recessive allele for white, it will still appear red?
That's right. Now we're going to talk about different types of dominances and how this occurs in heterozygotes.
Finally, something I get.
Incomplete dominance is when an organism resembles the phenotype of both parents. Now, in this one, a red flower is crossbreeded with a white one, and hence, since it's incomplete dominance, they will both merge together, and the color they will get is pink. The heterozygote will be pink. And codominance would be if the offspring shows equal distribution of both dominant and recessive phenotypes. So an example would be this, in which a red cow and a white cow would breed together and would create a rowan in which it would be blotches of white and red. So it's different from this because this is completely mixture, while this one is blotches, so it's codominance. Now, that's right. Mendelian genetics can be connected to evolution. So this can be connected to Darwin and evolution, in which all of these different variations can cause evolutionary change over time. Now the red and the white being connected together to get pink would cause an evolutionary change and new alleles for this phenotype of a pink flower. Hence the connection. That is all for today on Mendelian genetics. Tomorrow you will speak with Ms. Thymine on how genetic variation and genetic disorders and mutation is put into that.
Hello, my bio buddies. Miss Thymine here. Ever wonder what happens when there's an error in DNA replication?
Oh no, you mean something goes wrong?
Unfortunately, Angelique, nothing's perfect, and sometimes this can result in a mutation. There are several types of mutations. One type of mutation, called a point mutation, is an error in a single nucleotide of the sequence. Some point mutations include substitution, silent, missense, and nonsense mutations. Frameshift mutations include deletions and insertions.
Gee, that's a lot. What happens in substitution?
Well, this is when the DNA sequence contains an incorrect nucleotide in place of the correct nucleotide. It looks something like this. You can see that as a result of this mutation, a new amino acid is encoded for.
Wow, just one change in that teeny tiny sequence can do all that?
Yep, but don't worry, point mutations are usually not harmful. It's the frameshift mutations that can do the damage. Remember, frameshift mutations result from insertions and deletions. Who thinks they know what an insertion is?
It's when a nucleotide is added to the sequence and can cause the amino acids to change.
That's exactly right. And the deletion? Well, you'd be right, considering that part of your chromosome is actually lost during DNA replication. Essentially, you're omitting a nucleotide.
What happens in a silent mutation?
Isn't that when the new codon still codes for the same amino acid?
That's correct.
All this stuff about mutations are cool and all, but what the heck is a nonsense mutation? Sounds like nonsense to me.
Kids, nonsense mutations are not nonsense and should be taken seriously. This is when a codon for an amino acid changes to a stop codon, preventing translation altogether.
Okay, I got it.
Oh, Angie. Hey, doesn't a missense mutation cause sickle cell disease?
Indeed it can, because it can change the codon from one amino acid to that for another. Also remember, sickle cell is an example of pleiotropy. What is pleiotropy?
Pleiotropy is when a gene has multiple phenotypic effects, which is why sickle cell can cause a series of symptoms.
Thank you, Angelique. Well, this is a great segue into our topic of genetic disorders. Sickle cell, Tay-Sachs, and cystic fibrosis are just a few examples of autosomal recessive genetic disorders. As you can see from the pedigree for an autosomal recessive disease, the infected offspring is always homozygous recessive, and the allele for the recessive disease skips generations. This pedigree shows an autosomal disorder that had been passed down from two parents. In a pedigree, squares indicate males and circles indicate females. Any shape filled in indicates that the individual is infected. In this case, the infected male in this pedigree acquires the autosomal recessive trait.
What about autosomal dominant disorders?
Huntington's disease is an example of an autosomal dominant disorder, which could be fatal like most of these diseases. In order to inherit an autosomal dominant disorder, the infected offspring must possess one dominant allele. This often occurs at high frequencies and can also be represented in pedigrees. Does anyone know anything about non-disjunction?
And that's when I remember something evolutionary, something that our teacher, Miss C, had taught us about meiosis. If disjunction makes place in anaphase during meiosis, is it safe to say that non-disjunction is the cause of Down syndrome?
That is correct. Miss Cytosine would be so proud to know that you've remembered your phases. Down syndrome, or trisomy 21, causes the zygote to have 3 copies of chromosome 21 instead of the usual 2. Now, Angelique, could you give me an example of a sex-linked disease?
Oh my god, I'm blanking. I'm blind to this question.
That's absolutely right. Blindness, well, more specifically, color blindness, is a sex-linked disease.
Yeah, I knew that.
Now, could anyone tell me the requirements in order to inherit a sex-linked recessive disorder?
Well, the trait is carried onto the X chromosome, and males are more likely to be infected due to the inheritance of an X chromosome from their mother. However, males can never be carriers.
That is true. And we have a certain way to denote this inheritance, as shown here. But there is a special name for what we classify mental disorders, cancer, diabetes, and other autoimmune diseases.
Epigenetics.
Correct. Epigenetics allows us to change the phenotypes without changing the underlying DNA sequence, and passing these traits on to your offspring. Can anyone name the 3 mechanisms of epigenetics?
DNA methylation, histone acetylation, and
miRNA.
Good job, ladies. Another thing I forgot to mention was polygenetics, a type of genetic expression in which many genes influence the expression of a single phenotype. Examples include skin color, eye color, and height. Well, this concludes our discussion of genetics. It's been an absolute pleasure being one of your teachers. I hope you learned a lot.
Hey, Miss Cytosine here. Feel free to pause the video and answer this question on your own. Okay, even though it says 3, I will be walking through all 4. Okay, so connecticores. The location is the centromeres of condensed chromosomes, and the role of them is that they had the microtubule attachment sites, and without them, chromosome positioning and movement would not be possible. Microtubules are 100% necessary, and their role is that they find the axis of division and cytokinesis. Also, their location is in the spindle apparatus. Going on to motor proteins, they are located inside the eukaryotic cells, and they generate force to pinch a cell in two in the animal cell cleavage furrow. Lastly, actin filaments assemble under the membrane at the cytokinesis site and interact with the microtubules to position the spindle apparatus in the cell.
Using the given information, determine the correct order of the following events during the replication of the lagging strand. A. The DNA double helix unwinds. B. The Okazaki fragments are joined. C. The RNA polymerase builds an RNA primer on the parent strand. D. Nucleotides are added and matched to the parent strand. A is first because DNA double helix unwinds with the enzyme helicase. Then, the enzyme RNA polymerase builds an RNA primer on the parent strand to prepare for the new nucleotides being formed, which is choice B. Then, choice D, nucleotides are added and matched to the parent strand because of the RNA primer. And then it's choice B, the Okazaki fragments are joined by the enzyme DNA ligase.
A new mutation that arose in one copy of gene X in a somatic cell results in the formation of a tumor. Which of the following pieces of evidence best describes how the new mutation directly caused the tumor? A. Protein X normally stimulates cell division, and the mutation created an overreactive version of protein X. B. Protein X normally activates a growth hormone receptor, and the mutation decreased the stability of protein X. C. Protein X normally prevents passage through the cell cycle, and the mutation created an overreactive version of protein X. Or D. protein X normally regulates gene expression, and the mutation created an underreactive version of protein X that blocked the cell cycle? The answer is A, because somatic cells rapidly divide due to a signal that is sent by a protein, in this case, protein X.